spatial modeling. And today's talk will be on ancestry distribution models. Thank you.
So, hi, hello everybody. Thanks for inviting me to your seminar. So, yeah, so as I was, as we were saying, I'm a postdoc in multi-sliking lab, but today I'll talk about some work that I've done during my PhD in France. And so, I was very interested in the relationship between population genetic structure and environment, all kind of environment. And today I present specifically this model that we developed and that we call ancestral distribution models. And you will see why. And that we can use to forecast population genetic structure in case of some environmental change. And I'll show you what I've done on this data set of 20 species of alpine plants. And this is a work that I've been published last year in molecular ecology. So the idea is that there have been a lot of studies showing that you do find plants and other organisms that are sensitive to climate and especially to temperature. And you also have evidence for shifts that already occurred where species will move to keep track with the changing environment. And so what we wanted to, so usually what people are doing is that they will use species distribution models, which are models that are used to forecast the species distribution shift when the climate or environment is changing. So the basics about species distribution model is that they are correlative models. So they will use presence or absence information, so presence of an individual of a species, and they will correlate that to some environmental variables. And on this part, I'm only showing the temperature gradient. So they will use this information to infer the niche of a species. And then in case of change in temperature in this case, you can predict the new locations where these niche requirements are satisfied. So in this case, so here it's cold environment and warm environment. So in this case, if the temperature is increasing, and at the bottom your temperature are too high, but not at the top your temperature are not too cold anymore. So you will predict a shift of the species distribution toward the north. So the thing is that when you use this kind of model, you're not taking into account the genetic variation at all. So all the individuals were represented by these purple identical dots. But what we know is that in reality you have a lot of species where you can observe some population genetic differentiation. And so it might be that this different population would react differently to climate change. So the idea, I'm showing this really famous plot that you can obtain with structure. So where you have the species, but you have different clusters in the species, which are represented by different colors. And each vertical line corresponds to one individual, and one individual can have ancestry in one cluster or several clusters. And that's what we call ancestry coefficients. And so the idea here is to be able to forecast changes within a species, like among these genetic clusters that are different. So that's why we developed these ancestry distribution models. And very simply, what we wanted to do is to identify variables, environmental variables that were correlated to genetic ancestry instead of correlated to occurrence data. And then we'll be able to use that to predict these intraspecific changes. So the new framework is this. So for each individual, we have loci, and so we have neutral markers and maybe adaptive markers. And so in this case, we can observe population structure. So we have two ancestral populations, a green population and an orange population. And so you can infer, for each locus of an individual, you can infer the population of this locus. So either orange, either green. And if you look at this plot, you can see that there is a trend where individuals at the top have more green ancestry, whereas individuals at the bottom have more orange ancestry. So the population structure is linked, in this case, to this temperature gradient. And why is it so? So one explanation, and what is the explanation here, is that you have some loci that procure some adaptive advantages. So all the individuals that live in the cold cluster, they have this blue adaptive loci, whereas all individuals that live in the warm cluster have this red adaptive loci. So this blue line indicates the contact zone, which is permeable, right? You can have, obviously, you can have a lot of gene flow between these two populations. So the idea is that if you again have this increase in temperature, then the individuals that carry the red loci will be more able to move and to live in this warmer environment. And so you will see a gene flow so that individuals that live in this environment are able to, so that this environment is suitable for the individuals, and it's not suitable for this individual that carries a blue allele. So what you will predict is that you need to have some gene flow from the warm cluster to the cold cluster. And so the idea is that even if you don't have even if you don't observe this adaptive loci, you still observe all the neutral variation, which is usually linked to the adaptive variation. So you still have some information to predict those changes. So we developed this method that will, at the same time, infer the population structure and infer the correlation to the environmental variables. And so what kind of data we need are, of course, genotype data and environmental and maybe geographical data. So to give more insight about how the method is working, so the idea is that we have a lot of parameters and we don't know a lot of things about them. So for example, we don't know how many populations we have, we don't know the population of each individual and the population of each locus for each individual. So all this stuff we have to infer them. So the first idea is that the genotype of one individual at one locus the priority of this will depend on the population of this locus. And the ancestral population of a locus will depend on the ancestry coefficients of of the individual that carry this locus. And then the ancestry coefficients of this individual will in turn depend on environmental variables. And so that's where we include all this environmental and spatial information. So the idea is that these are the ancestry coefficients of the individuals, and we want to regress them on habitats, a spatial trend, and some autocorrelation. So this will allow us to infer the coefficients of this regression and thus the correlation between the population structure and the environment. So the idea is to infer all the parameters, so the allelic frequency, the populations, the regression coefficients, all these parameters at the same time in a MCMC algorithm. And so when you've done that, you can use the parameter that you just estimated for this regression, and you can use this regression to predict some ancestry coefficient on a map, for example. So you have a new location, you know the environmental data at this new location, then you can use your regression to predict the ancestry coefficient. And you can also use this regression to predict the ancestry coefficient in case of some environmental change. So basically you use your regression and you have the future ancestry that will depend on the future habitat, and then all this other stuff that you already estimated. So to summarize what the software is doing, so you're taking, so this is the entry, you're taking genetic data and environmental data, and then you estimate a bunch of parameters, and you can get the population structure of your individuals in your species. You can project this structure on the map, so each column corresponds to one cluster. You can also check some criteria to do model selection, and you can provide new environmental data. For example, data where all the temperatures have been increased by 2 degrees, and you can predict the new population structure, and you can also project this population structure on the map. And then what you can do is that you can compare the current population structure to the future population structure, and you can also compare the two maps. So we had a chance to have a really nice data set that was provided by this consortium, Intraspecific. And so in this data, so it's very nice first because we had 20 different species, all in the European Alps. But more importantly, what was very interesting is that they put a lot of effort in the sampling. So these are the European Alps, and they designed a grid on the Alps with all these small cells, and that were uniformly distributed on the grid. And in each cell, they tried to sample 3 different individuals, and this for each species. So this is really interesting because it prevents you from, this bias that you have usually in landscape when you do landscape genetics analysis, where you have some gap in the middle, and so you have 2 populations, and you don't know if it's because you miss some individuals or because you already have 2 populations. And so for each species, they sequence on average 150 loci, which are AFP. And so these are all the names of all the plants that are in the data set. So
where is this location?
so this is Italy right there, and then France. So it's European Alps. So in each cell of the grid, we also know the temperature, the radiation, precipitation, soil humidity, some topographic variables, and of course we do have the coordinates of the cell, so we know the latitude and the longitude.
What were the grid cell sizes?
What was, sorry?
The grid cell size?
So I think it was around 20 kilometers by 20 kilometers. So now that we have this method and this huge data set, we can ask several questions and try to answer to them. So the first question is, because we have this large number of species, we wanted to know if they exhibit common patterns of population structure. And then, of course, we wanted to know if this structure was correlated to the environment and especially to temperature, and then if we could find some evidence of adaptive variation. And if this is the case, assuming that you have some local adaptation, then try to predict the changes in population structure for different scenarios of global warming. So you could have up to 4 degrees of increase in the next century. And then we want to use this projection on the map to infer the gene migration that you need to keep track with the changing environment. So these are the results for 3 of the 20 species. So at the bottom right here, you have the ancestry coefficients that have been estimated for one of the species, and so you can project that on a map. So you have the red cluster and the blue cluster, and you can see that you have this climb of admixture coefficient between the 2 clusters, and so this is called the contact zone, where you probably have some gene flow between the 2 populations. And so what you can see is further the clusters are really well defined geographically. They're like all close, yeah, all individuals in the same cluster are close to each other. And then a very striking thing that we saw is that in 90% of the species, we found a cluster that was significantly warmer than the other clusters. And so this is the cluster, these are the clusters that are circled in red. So this was found for 90% of the species. And with each time this climb of admixture coefficients between the warm cluster and the cold cluster. So the question is, are these warm clusters locally adaptive or not? So obviously, before thinking about local adaptation, you should try to see if you could explain this cluster only using some neutral demographic processes. So let's say that all species experience the same let's say that all species experience the same demographic processes, then you will expect to get the same kind of population structure. And so you might find this correlation between environment and structure only by chance. And this is a likely assumption because we already know that the plants during Ice Age found this refugia and then so they found this refugia, and then after Ice Age you have this new expansion And so if all species form the same refrigerant, then you will have the exact same expansions for all species. But this could be easily checked by looking at the diversity. Because if you have some expansion, then you expect to see a gradient in diversity, and you expect that the source of your expansion will be indicated by your maximal diversity. So we can check the diversity patterns for each species. And so what you see here is two species that both have this warm cluster. And if you look at the pattern of diversity at the right, then you can see that they are completely different. So they don't, these two species at least don't share the exact same demographic history. So to go a bit further and investigate a bit more this demographic hypothesis, we did simulation of neutral expansion either from the southwest or from the northeast of the Alps. So this is supposed to be the Alps, and so we did some simulation of expansion this way or this way. And for that we used Splash, which is a really good software for simulation with especially explicit scenario. And you can use this map that is supposed to mimic the Alps. So you have this friction map, and that makes your simulation more realistic. So you can simulate some genetic data and then use the same method as before to infer the population structure. And then you can compare your simulated data to your empirical data. And for that you can use different statistics, and I will show only one of them, which is the correlation between your structure and your environment. And so if you look at this histogram, so this gives the level of association between genes and environment, and the black histogram are the values for the simulated data, whereas the color lines are the values for each of the species that we have. And so what you can see is the correlation between structure and environment is much larger for the data that we observe than for the simulation. So this makes us think that the neutral expansions fail to explain this high correlation. So if the demography is not enough to explain what we see, what about local adaptation? So we try to get a direct evidence for local adaptation, and for that we use this method that were proposed by Just and Conseil, and also co-pricing, which are methods to detect outlier loci. And this loci would be likely to be either adaptive, either linked to some adaptive loci. And so the idea is to look for loci that have allele frequency extremely linked to some environmental variables. And so we found some of this, we found outliers in some of the species. And so here I'm showing the allele frequency for outlier loci for 3 different species. And so if you plot the frequency at this loci as a function of the latitude, you can see this very, very sharp climb. And the interesting thing is that this climb corresponds to the contact zone between the warm and the cold cluster. So they are definitely associated with this warm cluster. And so here you can see the p-value that you can get or like the opposite opinion that you can get from this test. So we found evidence for this kind of potentially adaptive loci for all species that have this warm cluster. And as I was saying, they are associated with contact bonds. So the idea now is that because we think we have at least some hints for local adaptation, then we can go ahead and use our model to forecast changes within a species. And I mean, we need to look for local adaptation because the model makes sense only if you have local adaptation within the species. And so we looked at these different scenarios of global warming, and the most extreme is an increase in temperature by 4 degrees in one century. So because we estimated all these parameters in our model, we can now use them and forecast the ancestry coefficients for the warm cluster. So here it's the current ancestry coefficient projected on a map, and you can see that the contact zone is here. And so here you have the cold cluster that we are not showing. And for a 2 degree increase, you can predict the coefficient and then project again on the map. And you can see that the contact zone is shifting towards the northeast. And if you do that for 4 degrees, then it's shifting even more. So the fact that this contact zone is shifting would be due to some gene flow between the warm cluster and the cold cluster that you would need to keep track with this changing environment. And so the idea is that we wanted to measure for each species the magnitude of this shift. So it's very simple. You just, if you look at your cluster, you have your contact zone, and then you just measure along this transect. You measure the distance between the inferred contact zone and the predicted contact zone. So you can do that for each species. And so you can plot the y-axis is the magnitude of this shift, and the x-axis is the increase in temperature. And each line corresponds to one species. And so you can see that you have this global trend, so it's always positive, so you have this global trend of shift towards the northeast. But the magnitude of the shift is actually species specific. So you have some species for which the shift is really, really important, whereas some species almost don't show any shift. And so you can use these values. And so if you assume, for example, that you have an increase of 2 degrees in 1 century, then it would mean that you will need you will need some gene migration, and you will need this migration rate to be almost 1 kilometer per year to keep track with this changing environment. So you can look at a bunch of different statistics that will measure these changes, and I'm just showing another one, which is that we call intraspecific turnover. And basically, instead of focusing only on this contact zone, we are looking at the global population structure, and we are calculating the correlation between the current population structure and the predicted population structure. And so if this correlation is high, then it means that you don't have a lot of changes due to global warming, and so your turnover is low. if the correlation is low, then your turnover is high. So you can do the same as before and plot this correlation for each species as a function of the temperature. And what you see is that if you look at only a 2-degree increase, then your correlations on average stay higher than 65%. So meaning that the turnover is quite moderate. But now if you look at a higher increase, so if you look at 4 degree, then you can observe two modes. So you have basically half of the species that experience only a moderate turnover, and then half of the species for which the population structure will completely change, like, yeah, a lot. So just to summarize these results, so we can use the model to get some estimates about both the gene migration that you need to keep track with the environment and the global turnover that each species will experience. So now the thing is that obviously we had to do a lot of simplification for this model, and this will impact the way you have to interpret these numbers. So the first limitation that we have is that we didn't model interaction between species. So we assume that all over the range of the Alps, the interaction were the same, and we also assume that they would not change in time. So even if you have global warming, this interaction will always be the same, which might not be true. And if it's not true, then of course this would maybe slow down the migration of a population, because this population maybe will be adapted to the climate, but maybe will not be adapted to this species that you have in the future locations. And then we also assume that the global warming occurred in a very short time scale. So you don't have time for new mutation to arise and be adaptive and be selected. So you don't have time for that. But if this was the case, then you can imagine that the population that are in the cold environment, get this mutation and suddenly become adapted to the climate, and then of course this will also slow down the migration of the warm populations. And then last thing is that we didn't have phenotypes, so we cannot model phenotypic plasticity at all. And so all these things will because of all these things, then the gene migration rate that we have is actually an upper bound of the rate, and all these limitations are also actually all these limitations are also present in the species distribution model, so it's not only due to our model. So, as a conclusion, I'll say that yes, we did a lot of simplifications, and also we need to couple this model with some local adaptation scan. But I think we have some new perspective to include this genetic information in the species distribution model and use it for forecasting changes. I just want to thank my supervisor and all the people that work on this project in Grenoble and also in Switzerland, and of course the Intrabiodiff consortium that gave us the data. Thanks.
Excellent. Flora left us plenty of time for questions, so here we go.
This is really fun. But I was thinking, for those alleles that might be responsible for adaptations, debating, like, would those race ahead of, this front between these two types and sort of break down? Just since the migration rate of these alleles under selection versus neutral ones that might be giving you that signal or different rates?
So it will be really interesting to look at that, especially because you know that when you will have this migration, then you can have integration into the local population, and you can kind of, because you only need actually the adaptive loci, maybe you will use the other one. But it will be very interesting to look at that, but we don't have the data for that because we only have 150 loci. So I guess they're supposed to be neutral, so they're just kind of linked to some adaptive variation, but they're not themselves adaptive. So.
Can you use the same method to go back in time? So if you could have some maps of what this area looked like at different times in the past, and you knew something about the temperature in the past, could you compare the contemporary distributions and figure out
Yes,
the signal in the past?
We could do that.
Because
we don't have this data.
But it seems to me like you're comparing contemporary distributions to future ones, but the contemporary distributions also incorporate all the changes that have happened in the past as well.
Yes, and actually that's exactly the point, because they incorporate all the past changes, they also incorporate this local adaptation stuff. So I mean, at the beginning they were not adapted to the environment, and then you had these changes and they became adapted to the environment, and so by using the current data, we're also actually using the past information about this.
But depending on the dispersal rates of each species, then some of that is going to be more noisy than for different species than others.
And in trying to include the dispersal rate more, so you can say some stuff about this dispersal here. So for example, you can look at the migration and then look at your information about dispersal rate, and then say if it's likely or not, if you can make it or not. So you can do that, but it would be really nice to try to incorporate some more precise information in the model. Because right now, all species are considered, they can move the same way as well.
Maybe I missed, but are these all annual plants?
Yes.
That pops a whole
They were all
lot.
perennials, right?
Yeah.
Perennials.
No,
all
annuals.
perennials.
That's not
Sorry?
They were all perennials.
Oh,
They're
they're perennial.
Sorry.
Well, then that rate of change would be extremely rapid, wouldn't it? I mean,
Yeah.
be easy to understand for annual species, but if you look at all the competitive interactions and all those sorts of things, and something that moves almost a kilometer per year.
But a lot of these are could be wind pollinated. I mean, that's not a big deal for some species.
So it's really hard to estimate this long range
species as well.
dispersal. And I agree that maybe it's a lot, and also you have to remember that these are problems just because of all the stuff that we say that you can have, for example, introgression. Maybe you don't need your species to go as far, because if you just move a little bit, then you will have some allele surfing at the front of this expansion, and then you don't need to migrate as far as what we are saying. But this is really hard to do. I guess we could do it.
So on that topic, wouldn't you expect that species that show local adaptation would actually potentially have lower dispersal rates? And so that there would be some confounding of the degree of local adaptation with the dispersal rate, so that actually projecting migration might be quite complex.
So do you think this population will have a lower dispersal rate because
Because if they have a high dispersal rate, then dispersal could swamp out any local,
So you have some
locally
kind
adapted
of
alleles.
incompatibility between your two clusters that will reduce the diversity.
Either incompatibility or simply physical inability to disperse due to pollinator, just distances, genetic neighborhoods of some kind.
That's a good question. I've not seen that.
You'd sort of assume that the plants that are established there, I mean, the whole competitive relationship and new seeds coming in, becoming established and so on, I'm just surprised at how rapid the migration is.
Well, it's not that it's rapid, it's that the projection is that it has to be rapid.
So we're not saying
Okay.
this will happen, we're just saying if you want to keep track with
Yeah,
this,
okay.
then you have to
Good,
do
thanks.
that. You will have to move as fast.
But, I mean, sorry, one follow-up. Just the point is that you can have strong patterns of population structure that can result from really low dispersal, but it could also result from strong selection and local adaptation. So it's not. It could be either, and you couldn't ascribe one or the other just because you found strong patterns of
Right, but
population
it
structure.
could be confounded. You could have a
Right.
confounding of the degree of local adaptation and dispersal because those mechanisms do, that selection and migration would counter each other.
So I'm curious about whether you know of anyone doing a project that has sort of looked at historical genetic structure in a species, say, from a museum collection, and then collected modern data over the same distribution. And often we know how species have moved their distribution, but we might know how the genetic structures are different. But I'm wondering if you could use your method with the historical museum genetic data to try and separate out alternative hypotheses on how you arrived at what you have today. But to do that, I think you not only have to have climate data, you'd have to have landscape, like how landscape changed. You think you could use your method? So you could make projections of where it went and how quick it must have gone based on those parameters, and then you can look at what's actually observed today, sort of both ground truthing it, but also exploring all kinds of dispersal and landscape features. Is anybody trying to do that? I think you could do it.
I'm not sure. I'm not sure I know someone doing that. I guess you could. I feel like you could do it, but my concern with this model is that at the end, so you're saying you need this migration, or maybe there was this migration in the past, but you're not able to retrieve exactly the genetic, the actual genetic. The genotype that you had in the past. So I'm not sure
I think you
if
can retrieve the genotype.
But then we'll have to add some biological stuff in this model for sure. But maybe I'm misunderstanding.
My idea is if you scored the genotype, say if you're 150 loci from historical plant specimens that occurred around between 1900 and 1920, right? So you have that genotype data, and then you have the modern data. That would be and then you did all your predictions based on the 1920s data. It would be interesting to see how well it correlated with what you observed today, and your model seemed a way that you could tease out what might be really biologically important. Is it just the environment, or does it have to involve interactions because it didn't happen fast enough, or it happened faster than it?
Okay,
You see what I mean?
understood. Yes,
You could really much more finely test explicit hypotheses that is very difficult to do in any other way.
Yes, yeah, I understand. Yeah, that's true. But then it would be hard to say if there was more interaction or if there was more adaptation or new mutation. But yes, that's good.
How does the model deal with novel climatic conditions that weren't there in the current? So are you assuming that the warm cluster remains as locally adapted as it is in the present, even though you get the 2 degrees warmer, which wasn't available anywhere in the landscape in the first place?
Yeah, so that's a good question. So currently, but just because we didn't model it, but it would be, I think, quite easy to add it in the model. But currently we have this stuff where we're not saying anything about this part. I mean, this part is always red, but if we want to predict something here, then we would need to add some absence data. So like to know if the plant here is really absent, or if we just, because it's outside of the grid, is it absent, or is it just that nobody sampled it? But if we add this information, then yes, we could do what you're saying, and like a kind of species distribution model plus genetic distribution.
Any further questions for Flora? All right, let's thank her again for the very